To address neuroinflammatory conditions effectively, we have to stop looking at the human body as a collection of isolated biological organs. Instead, we need to map it as an integrated, systems-level hardware network. In this schematic, the gastrointestinal tract operates as the central logistics hub. It is the primary power station responsible for generating the biochemical current needed for neural signaling, while simultaneously breaking down the raw materials required to maintain the physical hardware. The stability of the brain depends on the absolute purity of the materials output by this hub. The entire network degrades when the internal logic of this logistics hub fails. This creates a state of supply chain contamination. Biologically, this failure often manifests as the uncontrolled fermentation of complex dietary proteins, specifically milk, casein, and gluten. When these proteins are not properly processed, they undergo uncontrolled spoilage inside the body, flooding the neural bus with intense metabolic noise. Because we cannot directly observe the internal processor state, we rely on clinical telemetry to measure this hardware-level failure. A primary tool for this is urinalysis, which tracks the specific metabolic exhaust produced by the gut. By analyzing this telemetry, we can calculate a microbiome readiness score, or MRS. This metric serves as a strict engineering prerequisite for safe system operation. Verifying the stability of the gut via the MRS is a mandatory pre-flight check before exposing the hardware to any high-energy impulse event like a vaccination or viral infection. To understand exactly how the gut fails, we can look at a commercial model of a controlled software installation, the industrial production of yogurt. To create yogurt, pasteurization acts as a system wipe, applying heat to kill off wild code, creating a clean disk. Next, high-fidelity bacterial drivers process the milk through a controlled fermentation loop, creating a highly compatible systemic superfood. Contrast this with natural spoiling. If raw milk is left to ferment without the system wipe or the proper bacterial inoculation, random bacteria compete for the substrate. The result is a logic conflict. Instead of yielding beneficial output, this uncontrolled breach produces toxic metabolites, putrefaction, and dirty biological current. This identical failure occurs in a compromised gut. A healthy system produces enzymes, like DPP-IV, to process complex proteins. When drivers are missing, the process halts. Undigested casein sits in the logistics hub as a protein-rich substrate. Pathogen nodes, like Clostridia, consume these intact proteins. In industrial fermentation, engineers use a casein-based Mueller-Miller medium specifically because it maximizes the toxin output of tetanus bacteria. Inside the human gut, milk acts as the exact same yield maximizer for related pathogen strains. Under these conditions, dairy provides zero nutritional benefit. It functions exclusively as high-octane fuel for the malicious code driving systemic neuroinflammation. This specific enzymatic failure leaves a quantifiable diagnostic signature in the body's exhaust. The urinalysis research conducted by K.L. Reichelt and A.M. Knivsberg provides the primary clinical documentation of this failure state. This chart shows the elevated spikes of exorphins found in the urine of autistic patients. The key markers identified are distinct peptide sequences like bowlide casein marsamd and glutamorphin. These intact peptide chains represent logic glitches. A functioning system would have dismantled them using peptidases, but here they have breached the digestion protocol entirely. The clinical correlation in this data is absolute. Every single patient in the sampled autistic subset showed an increase in one or more of these specific dietary opioids. This telemetry confirms a hard structural breakdown in the logistics hub. Proceeding with recovery requires a mandatory logistics lockdown, achieved by removing all gluten and casein from the supply chain. Uncontrolled fermentation creates massive localized inflammation. As the gut lining swells, the logistics gate, the intestinal barrier, physically degrades, pulling apart the tight junctions. This creates a leaky gate scenario. Highly reactive molecules like exorphins, alongside bacterial lipopolysaccharides, or LPS, spill directly into the systemic bus of the bloodstream. In circulation, LPS acts as electromagnetic interference. It penetrates the blood-brain barrier, triggering the microglia, forcing them into an M1 attack state. This continuous high-voltage threat signal locks the microglia into an aggressive defensive posture, halting maintenance functions like synaptic pruning. This continuous feed of gut-driven dirty current explains why severe neuroinflammatory behaviors persist long after an initial biological trigger, like an illness, has cleared. The supply chain failure creates a hysteresis latch. The brain is locked in a permanent, hardware-level state of inflammation, driven entirely by the damaged logistics hub below it. Attempting to treat the system while the gut is in active dysbiosis carries immense risk. The continuous injection of LPS means the processor operates with zero-voltage headroom. It is already running at peak capacity. Biological impulse events, such as administering a vaccine or contracting a viral infection, act as massive high-energy inputs to the immune system. When one of these high-energy impulses hits a system already saturated with LPS and exorphins, the dampening capacity is exceeded. The voltage spikes past the ceiling, causing a monster wave crash of extreme signal clipping and behavioral regression. To prevent this catastrophic surge, engineers must utilize the microbiome readiness score to assess baseline stability. The protocol tracks 3 metrics. The first is Bristol stool type. Formed, type 3 or 4 output proves basic digestive capacity and contributes 4 points to the score. The second metric is the VOC audit. Confirming sweat and exhaust maintain a neutral odor ensures no active ammonia putrefaction, contributing 3 points. The final metric measures structural jitter. A calm abdomen confirms the system isn't generating biofilm fuel, bringing the total score to 10. A score of 9 to 10 indicates a green state. Only at this threshold is the system cleared to process an impulse event or initiate an active medical flush. If the baseline score is below a 7, the hardware has no headroom for additional stress. All medical titration must stop. The lead engineer must focus entirely on sealing the logistics gate and applying coolant to lower the inflammation. By utilizing telemetry to calculate readiness, we replace biological guesswork with hard engineering logic. We verify the foundation is stable before we apply a load.